I'm delighted today to be speaking with Dr. Penny Cristanacci, an MR physicist and MR Core Lab lead for NCITA, and also with Professor James O'Connor, a professor of radiology. And both your research focuses in quantitative MRI. So to start with James, what is an imaging biomarker?
So a biomarker is a defined characteristic that can be measured, and it can be used as an indicator of normal biology, of pathology, or of response to therapy. And an imaging biomarker is where we can use an imaging method such as PET or CT or MRI to derive a measurement.
So when we think about the clinical translation of MR biomarkers, to what extent are there unmet needs within this area?
So there are many unmet needs. There are a large number of MR biomarkers out there, and many of them have had use in the clinic, but far too many get stuck at the stage of being a scientific idea and don't actually benefit patients. Now, part of the reason for this is we don't validate the biomarkers. Now, validation can be thought of as a multi-step process where we start to get more and more evidence and understanding of how a biomarker performs. And the various steps can be technical or look at the biology and the clinical performance. And some of the key technical steps are around biomarker standardization and harmonization. And that's where the MR Core Lab comes in.
So what can we do about this issue?
So one of the things we are doing about it in the UK cancer imaging community is setting up an infrastructure with NCITA. And there are various aspects that we're trying to address around technical validation of MR biomarkers, in this case particularly in cancer. And one of those is the role of an MR Core Lab to improve the way we acquire and analyse MR biomarker data.
And Penny, what are your thoughts here?
So we are developing MR Core Lab within NCITA, as James said, in order to improve the quality management of quantitative MRI biomarkers and aid their clinical translation. So this is a unique and UK-wide imaging infrastructure. And its support is MR method independent, so it doesn't matter what method your study is using, and it's integrated into the NCITA XNAT repository, so we can also support the data sharing and storage requirements of a study.
Fantastic, thank you. And of course, there are a variety of studies taking place across the UK that will all need different levels of support, likely. How can the NCITA MR Core Lab really provide the flexibility to support this varying level of need?
So we're providing 3 levels of service: a basic, intermediate and advanced. The basic level of service involves providing a lockdown imaging protocol. So we have scanner evaluation in order to ensure that the scanners can perform the imaging protocol that's required for the study or trial. Scanner qualification to ensure that it can do that to the standard required, and periodic requalification in order to ensure that it can do that for the duration of the study. The intermediate level of service adds to this data quality control of the live study data. We're checking that the quality of the data is good enough to go into the next stage of analysis, and we do this by using our integrated DICOM protocol checker to check that the data was acquired as per the lockdown imaging protocol, and using a series of visual imaging metrics to ensure that the data quality is good enough. And finally, our advanced level of service. This adds integrated data analysis. By containerizing code and running it through the XNAT platform, we can provide an end-to-end service from scanner evaluation right through to your study results.
Okay, thank you. So James, as a primary investigator yourself, what do you think the benefits are for PIs in using the MR Core Lab?
So the benefits can be substantial. A lot of the time investigators want to use imaging, but it's done in a suboptimal manner. And the benefit of using a core lab is that the data can be acquired and analysed in the best possible standards in a way that is akin to the quality seen in other non-imaging biomarkers.
Thank you. And what are the benefits for the site staff who are performing the imaging?
So the benefits for the site staff is they have some clearly defined protocols and manuals to adhere to, and this helps them acquire the data to the best possible standard. They're also able to interact with the other sites and with the central analysis team in the core lab, and collectively that gives better communication, and that in the end ensures the data is of the best possible quality.
Okay, fantastic, thank you. And Penny, what type of studies are supported by the MR Core Lab?
So NCITA's focus is on clinical research studies in the area of oncology, but MR Core Lab can support studies in any disease area. So a subject matter expert in academia who has developed a novel imaging biomarker, but in a single centre, but is yet to translate that into multiple centres and test its reproducibility, they could use the MR Core Lab. Clinical study teams who perhaps are using an established biomarker but in a novel disease area, they may not have the MR expertise within their team, they could use the Core Lab service too. And finally, because we're using our quality management system QPulse in order to monitor studies end to end, we can also provide a fully auditable trail of the study. So pharmaceutical-led studies and clinical trials that are using quantitative MR imaging biomarkers as primary or secondary endpoints could also benefit from MR Core Lab services.
Fantastic, thank you. Okay, final question then, Penny. If there are study team staff and clinicians interested in using the MR Core Lab service, what do they do? How can they contact you?
So go to the NCITA website, that's ncita.org.uk, and you can find out lots more information about the MR Core Lab services, and you can also find our contact details. And I really encourage people to get in touch as soon as possible in order to discuss how we can work together in order to make sure we get the best quality data from your study or trial, and also ensure that it makes the biggest clinical impact.
Fantastic. Well, thank you very much, Penny. Thank you, James.
Thank you.